introduce the final speaker in this session, Irina Antonijevic, who's the Chief Medical Officer of Triplet Therapeutics. Irina.
Thank you very much, Sarah. Good morning. Thank you very much to the EHDN committee for inviting Triplet to give an update on our development program of an MSH3 lowering antisense oligonucleotide for HD and other repeat expansion disorders. So my talk will be a little bit different. I will have less data, but I want to give you an update on Triplet, and as part of today's talk, I might be making forward-looking statements, so this is my disclaimer. So this is the outline, and I want to briefly go over the standard drug development process, and you have heard information from the previous speakers who have outlined particularly some of the issues with the risk of drug development programs and how many fail even at a late stage, as pointed out by Michael Hayden. So a standard drug development process entails some of these, I don't know, how can I, oh yeah, here. Some of these different phases. There's more iteration. It's not as linear as it looks here on the slide. But bottom line is that typically we start with finding the right target, the right molecular target, then developing a molecule that engages with the target in the intended way. And this can take, you heard this also from Amy Lee, can take several years to develop an optimal molecule. Then you do the preclinical testing and ultimately the clinical testing. Now, as we think of a rare disease, such as Huntington's disease, there can be an abbreviated path. So we can sometimes have very good data in a phase 1-A combined trial with a good proof of concept that can lead to accelerated approval. But at Triplet, we had a particular advantage because Triplet was founded based on the genome-wide association studies from Jim Guzella and Marcy McDonald and Youngmin Lee that told us that there were these genetic modifiers that had a very strong impact on HD onset and progression by modulating somatic expansion. So this led to the foundation of Triplet, and so we basically could jumpstart the drug development process by picking one of those genes that were strong genetic modifiers. But then, of course, we still had to do the exploratory toxicology and the IND-enabling toxicology, but we also hoped to do a combined phase 1-A study for an accelerated approval, potentially. Now, because we selected an antisense oligonucleotide approach, which typically leads to a more rapid path towards the clinic because there's a shorter molecular optimization phase, but still I want to highlight some of the key differences of an antisense approach that uses MSH3 as a target compared to mutant HTT. So what you can see here in the slide, and some of you might have seen this before, is that by lowering MSH3, we actually stop somatic expansion. So we don't have the question about wild type or mutant HTT, but we stop somatic expansion, and so there is no further expansion and thereby no further increase in the toxicity of the mutant HTT RNA and protein. And we know that this stopping of somatic expansion can be achieved at only 50% lowering of MSH3. Vanessa Vila had presented some of this data in mice. Triplet has also generated data in vitro in patient-derived cells, but also in animal studies. And because we believe that an unconjugated, so just a naked antisense oligonucleotide injected into the CSF does not reach the striatum when it's injected intrathecally at sufficient concentrations, we have decided to inject our ASO into one of the lateral ventricles in the brain, so intracerebroventricularly, because with this approach, we first of all think that it is more sustainable long-term for the patient population, but we also see that this secures adequate distribution to the striatum to achieve 50% lowering of MSH3. And because MSH3 has not been associated with any neurodevelopment issues, there is no concern in terms of the age when we can start to treat individuals. Now, Triplet was founded in late 2019, and because of the genetic modifier data, we were able to quickly select our target. We selected MSH3 in early 2020, and with the subsequent data, we still feel very strongly that this is a very relevant target for halting somatic expansion. We selected an antisense oligonucleotide, and because there was a rapid opportunity to develop this into the clinic, we were able to pick TTX-3360 based on multiple in vivo animal data, both in mice, rats, but also non-human primates, as a safe or at least well-tolerated antisense oligonucleotide. We have then started the IND-enabling studies in late 2020, and as you all remember, and we have heard earlier, in March of 2021, the Roche announcement came out that paused tominersen treatment, and then we heard the WAVE announcement of 2 clinical trials lowering HTT or mutant HTT that were discontinued. So at that time, Triplet has been in active fundraising. We had convened a great group of investors. As this data came out, these investors got cold feet, and instead of giving us the money that we were seeking, because we wanted to develop our lead molecule, but we also had promising backup molecules that we wanted to develop in parallel. So bottom line was that because of the overall concern with ASOs, and particularly ASOs and HD, we had to trim down the program, and so we used the funding that we had to progress TTX-3360 towards the clinic. So this meant that we invested in drug product manufacturing, so obviously we needed to have a drug ready, going through all the quality steps. We started to develop a phase 1 protocol with many wonderful advisors as well as patient advisory boards. We, of course, also had our natural history study, SHIELD HD, running that informed really very importantly the phase 1 protocol, and we had very good constructive discussions with regulators in Europe and North America. But as then the GLP tox study data came out first in the mid of the second quarter this year, and then the last data set just about 4 weeks ago, we have seen that there was some minimal adversity at therapeutic dose levels. We have done GLP tox studies in both non-human primates, so in monkeys as well as rats, and the adversity was dose dependent. And obviously a tox study is designed to show toxicity, so this is the goal of it. And dose-dependent toxicity is encouraging because if you can select a lower dose and achieve enough target engagement, this might be just fine. And when I say minimal adversity, this is a term used by the pathologists who assessed the data. But given, and this is maybe not a scientific argument, but it is nevertheless reality for a small company like Triplet, I mean, market influences do play a major role. And so with this data that made the phase 1 study a little bit more difficult, we felt that it would be best to partner our program with a larger company to help us look at both the clinical lead, but then also the backup molecules. But I do want to stress that we have a very specific adversity by our molecule, but we have no inflammatory response, there is no microglia response, there is no cytokine release, there is no NFL increase, and we have a very linear and predictable PK-PD relationship. So overall, there are certain really unique aspects about TTX-3360. But we also have developed backup molecules that, as I said before, we could not develop in non-human primates, so we have only in vitro data and some in vivo data in mice and rats. But these molecules were developed specifically with a greater potency in mind. We wanted to change the backbone structure of the antisense oligonucleotide in a way that we would be able to induce the same knockdown with less ASO administered. And you can see here, based on the in vitro profile, that we have, for some of those molecules, a much higher potency, which we think is very encouraging. So in summary, first of all, as of 2 days ago, I'm very glad to be able to announce that SHIELD HD has transitioned to CHDI, and we wanted to make sure that all the data in SHIELD HD that we think are highly, highly valuable to the HD community is in good hands and can be used to really help the community to develop drugs in HD. So I want to thank really everyone involved, but particularly, of course, CHDI. We have developed a target engagement biomarker assay to measure MSH3 mRNA in the CSF of humans, but also non-human primates. So again, this is a tool that we think is very valuable. And in this context, we have also developed MSH3 antibodies because sometimes you also want to measure the protein, not only the RNA. So this is also in our toolbox. We have the GLP tox study data with TTX-3360, and I do want to stress that none of the data that I've seen discourages me from MSH3 as a very, very important target to affect somatic instability. In fact, I really think MSH3 is a prime target when one wants to stop somatic instability. I also want to stress that all of the data we have, there is no concern with an ICV route of administration. None of the adversity that we have seen is linked to the route of administration. And as I said, in the current environment, Triplet is looking for a partner to, take on the lead, but also the backup programs, particularly given that there is a much higher potency for some of our backup ASO molecules. And with this, I come to an end. I want to thank the Triplet team. The Triplet team has been a highly dedicated team working to develop all these tools. I do want to thank many, many, many advisors, including people with HD who have given us wonderful advice to really help shape our phase 1 program, but also help shape the entire preclinical program. And I do want to thank CHDI. I mean, we have worked really quite tirelessly in the last few weeks to secure, actually months, to secure the transition of SHIELD HD to CHDI. And I take questions. Thank you for your attention.
Thank you, Irina. And I think Irina's talk also highlights the incredible challenges of drug development and the challenges particularly in a rare disease. So I think we need to applaud all of our speakers this morning because they're going down this path to try and help us with potential treatments for Huntington's disease. And I think, Irina, you really highlight the challenges of drug development for a rare disease. So we have time for one quick question for Irina and then the panel discussion. Question in the front row. The man with the pink shirt.
It's black. Hi, thank you. Ed Wild, UCL. It's not a question, but I really want to say that I think that the community really appreciates the openness and honesty, Irina. This is a difficult presentation for you to give. Triplet's worked extremely hard. MSH3 is an extremely promising target. ASO drugs, broadly speaking, are extremely promising and have proven themselves to be effective in a number of conditions. And the serial bad news from HD clinical trials last year, was it last year? It feels like 1,000 years ago, really has given investors who often have only a relatively superficial understanding of the complexities of our field, cold feet, as you described it. And that's a real shame. And I think Triplet are to be commended for their persistence and for their commitment, and also for their openness in sharing the current state of play, which I know, I think we're a community that appreciates openness and news of all kinds, and I think we are sufficiently mature enough to be able to cope with news that's not great as well as news that is, and a sense of the complexities and difficulties. So thank you.
Thank you.